Hey everyone, my name's Nate. I'm a PhD student in David Baker's lab at the University of Washington at the Institute for Protein Design. I'm going to be talking about a few things today. And if anyone has questions, please put them in the chat. And Hannes, I'm sure you will have questions, so stop me at any point. There's a few things I wanted to talk about. The first is this framework we've built in our group called AtomWorks, which essentially underlies most of the models we train. I then want to talk about two models that we've trained with AtomWorks. One is RosettaFold 3, which is an open source structure prediction model in the style of AlphaFold 3. And the second one is RF diffusion 3. I'm not going to talk in a huge amount of detail about RFD3, but I kind of want to describe what we did, and then it might be a future topic for a reading group conversation with some of the other folks involved in that effort. So these are the three high-level topics I'll go through, but absolutely jump in at any point with questions. I'd like it to be as much of a discussion as possible. And then after I talk about two or in the middle, I think it would be a good time to think about together what the gaps might be to uh, AF3 and the alternative reproductions. I think we have some thoughts there. So I'll start with what AtomWorks is. And to understand what it is and why it came about, we jump back to last year when AlphaFold 3 was released. And so in our group, when AlphaFold 3 was released a little more than a year ago now, we knew that this was going to fundamentally change the way we design proteins. So previously, the way we designed proteins for function at the Institute for Protein Design was the pipeline you see here, where we would essentially choose a motif in the broadest sense of the term. We would then scaffold that motif. We would use a tool like RF diffusion 2 for this. We would fit a sequence into that backbone. This is ligand MPNN. We would use a structure prediction filter to see if we recapitulate the designed protein after folding. RF all atom was one of the first all atom methods to do this. And then ultimately, we would sometimes have additional filters after the fact. And placer, also known as ChemNet, is what I'm showing here. So we have the point I want to make is we have all of these different tools, each of which were completely siloed from each other and developed entirely independently. When AF3 was released, it was fairly obvious that this would have implications on all of the tools that we developed in the lab. And it began to become quite clear that we should spend a significant amount of time developing a framework that would enable us to rapidly develop and iterate on these tools and allow innovations or improvements in one tool to expand to others rather than existing in isolated code bases, which is how they existed previously. So that's how we ended up with AtomWorks, which is essentially a biomolecular modeling framework that underlies all of the tools and the models released from our lab. This is just a representation of that. We have AtomWorks, which is the layer that goes all the way from the point of dataset curation and data ingestion through model featurization. And then we have the PyTorch models built on top. So that's RFD3, ligand MPNN, RF3, and any additional models such as filtering tools that we may develop in the future. The neat thing about this is now all of our models share probably 80% of their code. So AtomWorks is the vast majority of the code through all of these models, and then we can essentially swap out the PyTorch models on top, and we can quickly iterate and test ideas. To more pictorially describe what this looks like and why this is useful, this is an example of the featurization pipelines for some of the different models in our group, MPNN, RFL Atom, RF3, and RFD3. And what we're trying to show here is that there's common operations and common transforms that these models can share. So it's not only the literal data loading and cleaning piece, which is a huge component, but it's also the operations themselves. So things like cropping and generating reference conformers are fairly annoying things to do, but we've set it up such that we can code that as a transform, and then we can use that transform across any code base. Similarly, for loading MSAs, pairing MSAs, generating chiral features, these are generally useful operations. And the way that we designed this framework is such that we built them in a scalable, well-documented way such that we can plug and play in the future. And we've already extended this to other models in our group. For example, there's this new suite of partially latent, C alpha latent models. We can just grab the PyTorch components of those and connect them to AtomWorks and train models off the shelf, which has been super exciting that we can iterate so quickly. And then this is just kind of a final plug. We tried to build it as an open source library. We've lucky to have the support of some of the folks at Rosetta Commons who are building documentation for these tools and helping to maintain them. And we're excited to continue to expand this in the future. And so it enables you to do cool things like put breakpoints in the middle of a pipeline and visualize what you're looking at. So that's what the examples are here. It's you can grab your data loading pipeline, throw a breakpoint, and say what exists in my structure at this point, pre or post cropping. Here is a heme cropped to the pocket atoms. You can visualize annotations. On the right, I just got rid of the heme and looked at just the pocket. And so it enables a very powerful way of training models and even more broadly bioinformatics tools that are useful to people outside machine learning researchers in our group. That is all I wanted to say on AtomWorks, just sort of a brief plug for it. Any questions on that component before I jump into the modeling bits?
Yeah, does this mean we can now have, for example, libraries where we no longer need to switch between running RF diffusion and then protein MPNN and then Rosetta Fold 3, all with different repositories, but now we can have one repository, for example?
That's correct. So one thing we're excited about, broadly speaking, is inference time search. And it can be difficult to do that when each of these tools live in different repositories. But what we've set up with now in our group is we have all of the tools in one repository and they can all pip install each other. And so you can essentially do really neat cycles between MPNN and folding, and then back to diffusion, or even if operations are differentiable, trace gradients back through all of them to update your priors. And so it's exciting for us now that they're able to work together so well.
Yes.
I think that was one of the main motivations for this work too, is that uh, we have these complex pipelines that determine whether or not we actually order a design. And it would be nice if we could find a way of integrating these pipelines rather than chaining a bunch of scripts together, such that we can directly optimize on the objectives that actually decide whether or not we order a design. Because while we can certainly improve the filtering metrics we use, we want our models to maximally pass the metrics we ultimately use, which tend to be quite downstream of simply refolding.
Then about the inference time search, should we talk about that now or maybe in the third part?
Sure. I mean, I don't have any slides on it because this is very new, but we can talk. I'm happy. Let's do it after I talk about RFD3.
Yeah.
All right, I'll jump into Rosetta Fold 3. So nothing incredibly novel here. Rosetta Fold 3 is another AlphaFold-like model. There's others that exist. Uh, so I'm not going to dwell too much on the performance, but we see reasonably strong performance across the board, but a persistent gap to AlphaFold 3 when we benchmark all other models available as well, not only the open source models, the truly open source models like Boltz, but pretty much all the models, we see this gap. Kind of independent of what people say in their papers. Once when you benchmark them on recent PDB and D-League sets, AlphaFold performs the best by some margin on some tasks across the board. And so I think it's interesting question we can revisit at the end of why that is the case. I wanted to note that the model we released, RF3, we have a significantly better model in-house now. And to Hannes's point, like what makes the difference? For us, it's been distillation data. So we essentially added distillation data for Magnify, and you see the improvements across the board on all of the tasks. We've been quite surprised by this. Like the fact that we were able to improve protein only best of one above 0.82 to kind of closer to 0.83 was quite surprising because we're now at essentially AlphaFold level protein only performance by adding these additional distillation data sets. And this is only a small fraction of the distillation data that AlphaFold 3 is trained on. So in terms of what is the gap, this analysis suggests that distillation is at least a significant component of the gap.
So how is this distillation data generated here?
So this is magnified metagenomic sequences that are clustered and then predicted with Rosetta Fold 3 and then trained on at 50% probability during training. They're
Or
monomers.
okay. So you use 50% of the time you use this distillation data for fine-tuning, I suppose.
Training
Uh,
from scratch.
uh,
During fine tuning, it doesn't really help.
oh, cool. That's super cool. And how do you choose whether like you have some PLDT cutoff or so
Right.
for
Yeah.
monomers?
They're pretty much all highly confident predictions because these tend to have deep MSAs and they're fairly simple protein monomers. So something like 90% of 95% of them are predicted with quite high confidence. But we do indeed keep only the highest confidence ones and train on them.
What does the bar versus no bar here mean?
So this is, yeah, the plot is poorly labeled because this is work in progress. It's best of one versus best of five. So the solid color is best of one, the striped color is best of five.
Okay. Then it is kind of very curious how I assume in this distillation data you have no small molecules, right?
This distillation data has no small molecules.
Yeah.
Yeah, so why is
No
protein?
protein DNA and no and
Yeah.
also no RNA.
So the protein DNA and protein and RNA only had distillation, does have distillation. We had distillation in the prior model, but we added it in fine tuning, whereas this was added training from scratch. So protein only had new distillation data, and protein DNA and RNA only had DNA had distillation data that was included in RF3, but not only in fine tuning, not when training from scratch. So it's a pretty significant difference we see when adding distillation data, training from scratch versus in fine tuning.
Okay. So the change here, you think it doesn't like you already had these other previous distillation sets, right? And here the major change between RF3 and run 10 comes from the fact that you're using um, distillation data when training from scratch and not just fine
Yeah.
tuning.
There's two differences. The first difference is for protein DNA and RNA, we use the distillation data training from scratch. We also added 15 million protein monomer datas, distillation points. So those are the two changes, and it seems to help quite a lot, which is surprising. But we added no protein ligand or ligand only
Mm.
distillation data, but we still see improvements on those tasks. It's possible that predicting the protein better makes it easier to dock a small molecule. I think that's a reasonable thing to assume. Um, yeah.
Is your best of five evaluation here using the confidence model or
No,
the
this is just taking the absolute best of five. Our confidence module is not fully trained yet.
yeah, very cool. I think that's yeah, maybe one step toward the AlphaFold 3 gap closing.
Yeah, in terms of what are the other gaps, I think I mean, there's a few places it could be. So one is clearly the data, the distillation data. The second is the curation of the data. Everyone sort of uses the AlphaFold weighting schemes, but no one has the AlphaFold training data sets. And so it's very likely that the way we're sampling the data is suboptimal, in terms of how we weight protein ligands versus protein versus protein DNA. The actual way we draw samples during training is hugely impactful on the performance, but we are almost certainly not sampling optimally. It's just a difficult problem to tackle with finite compute because I don't really know how you search that search space of sampling hyperparameters in any sort of computationally efficient way.
Okay. Yeah, well, we also know, or in AlphaFold 3, they show these nice curves where, for example, for some antibody prediction accuracy, you can do a lot of inference time scaling by just sampling more and sampling diversely with different models, with different seeds and different MSAs. And then the confidence model is able to pick out good samples there. And now you, for example, see here this protein ligand gap being bigger in the top five than in the top one. Same for protein DNA. And then, yeah, it's maybe another gap of the public model to the um,
Yeah,
another gap.
our confidence model is definitely worse than AlphaFold's, if that's what you're suggesting.
Yeah. Yeah. Why do you think that is? Do you co-train the confidence model with
We do
the
not. We do the thing where we freeze the weights and we train the confidence module separately. It's possible that is the difference because that's the only fundamental difference
yeah.
that occurs in terms of training to our knowledge. It's sort of mysterious why there would be other differences. So their model sees way more optimizer steps because it co-trains for so long. So perhaps that could explain some of the delta.
Yeah, maybe there's sort of a regularization in that at the beginning of training, the confidence model sees bad structures or like it sees more bad predictions and then has more negatives or sees more negatives during training, and that then prevents it from being like overconfident in the correctness of predictions at the end of training or something like that.
Yeah, it's sort of the challenge because our trading loss is so low by the point we start trading the confidence model that it very rarely sees examples it does poorly on. All right. So this is, I think, one interesting finding that we've seen recently. I wanted to now touch briefly on two problems that we've seen with AlphaFold 3 in our group and some steps we took to address them. So one problem that is quite recurrent is adherence to chirality. We want oftentimes to predict chiral molecules, especially for things like enzyme design, where we want stereo-specific active sites and stereo-specific reactions. This is something which we care quite a bit about. We also see limited adherence to templating. And so we sometimes want to template small molecules, for example, a particular transition state. We sometimes want to template DNA or RNA, and quite often proteins, especially if we don't want to provide an MSA. And the ways of doing that for us have been quite challenging because the templates have been protein only for the most part, and they've also been homology-based templates as opposed to rigid templates. So we wanted to bake in at train time some additional tools that we were able to control at inference that would enable us to perform these two tasks. So I'll talk a little bit about the way we formulated chirality, and then I'll jump over to the templating world. So for chirality, in AlphaFold 3, chirality is given. It's only given through the embedding of what's called the reference conformer, which is for every small molecule and residue. They generate with RDKit a sample conformer, and they embed those coordinates directly to the model. And they also embed the pairwise distances directly to the model. This, if you think about it, very implicitly provides the chirality of the molecule, but it turns out it's not adhered to very well. The adherence is quite poor. So we wanted to see if during training we could bake into the model a form of chirality that would be better attended to and ultimately result in stronger chiral performance. So the way that we did that was at every denoising step of the diffusion module, we calculated the chiral angle. So there's a dihedral angle that uniquely determines the chirality of a given stereocenter. We calculate that angle during every denoising step, and we calculate that angle, the loss of that angle with respect to the ideal angle for that particular chirality, be it R or S, and we provide that as a feature to the model, the error of that dihedral angle. The reason we do this is something like R or S is extremely unnatural for a model to learn. If I embedded R or S, the model would have to implicitly learn how to order all of the atoms around a stereocenter and determine whether or not moving an atom would change whether or not that order was R or S, so clockwise or counterclockwise, which is extremely unnatural thing. I mean, in theory, it is possible neural networks are universal function approximators, but it would be certainly very difficult. So instead, we thought that this approach, by embedding the vector of the atoms, or the gradient of the chiral error with respect to the atoms, would essentially tell the model which way do I need to move these atoms in order to make the stereocenter conform to the correct chirality, which is a much more natural way of being able to embed this information.
So in AlphaFold 3, they also do not feed in the just a chiral feature, right? They only feed it in via the conform.
Only the conformer correct.
So have you tried what happens if you just feed in the chirality feature as opposed to your geometric chirality feature?
Using the reference do you mean feed it in only as the reference conformer?
No, you just give a binary feature to that token.
An RS token. So we tried that in Rosetta Fold 2 all atom. We did not try that in this case because we, from our previous ablations, found that the token feature was adhered to very poorly and basically made no difference. So we directly went to the geometric chirality feature.
So in RF diffusion 2, you found a clear difference between just feeding in the chirality as a binary feature versus specifying it with this vector.
Yeah, Rosetta Fold all atom, but yes, it was a huge difference. So we had that learning from before, and so we just directly applied that.
And you literally feed in this vector as a
Yeah,
so non okay.
yeah. So essentially, what we do is we compute this dihedral in the noisy coordinates, and then we compare that to the dihedral in the ground truth or the ideal dihedral for that particular stereoisomer. We then take the gradient of that error with respect to the atoms, and we provide that as a vector feature to the model. So literally an XYZ feature. And so it is quite literally telling the model which way do I need to move the atoms around this stereo center in order to minimize this chiral loss. And so we take this gradient only for the purposes, not for the purposes of backpropagation, but for the purposes of providing a feature to the model. And so it's a form of arbitrary conditioning, essentially, that we developed in Rosetta Fold 2 Alatum that has been applied here again for chirality, and one could imagine other use cases for it as well. We haven't explored applications outside of chirality, but for us, it's been reasonably effective. The other thing we did is we explicitly mirror the PDB with some probability. So if you have a given PDB structure, that's what this picture on the left is, you can mirror it across a plane. And now you end up with the inverse chirality of all of the residues in that structure. And we do, with some small probability, mirror these structures, which is a way of augmenting our data set and trying to encourage the model to actually attend to the chiral features we're providing it. Because otherwise, what we found is that the model essentially memorizes common patterns of chirality and doesn't adhere to the features that try to encourage the model to choose one chirality over the other. The results, all of this is that we find that on our test set, we end up performing better than all models on chirality. The other interesting piece to note is for bespoke applications, because we went through this whole song and dance because there are actual applications where this matters. One example of this are macrocyclic peptides, which are an important therapeutic class of molecules. In particular, mixed chirality macrocyclic peptides are interesting because they can bind diverse targets, but they also can be administered orally, which makes them exciting as a therapeutic modality. The models do quite poorly at predicting these mixed chirality L&D peptides, as you can imagine. But we found that with these augmentations we performed, we're able to significantly improve the performance. So on the left is backbone RMSD. RF3 has the best performance across all models. And then on the right is chiral accuracy. Here we ran bolts with guidance, so they got 100%, but that came at the cost of significantly worse backbone accuracy. And so we find that we've been able to maintain backbone accuracy while also increasing or better modeling the chirality.
Hmm. So these, yeah, this is very interesting. These mixed chirality peptides. I was aware of like completely swapped chirality peptides or cyclic peptides, but yeah, mixed sounds very interesting. So
Everything under the sun exists on us. One thing I've learned since trading structure prediction models is that there's a there's use cases for everything that one could imagine and then some. If you think something is too specific, it is never too specific.
yeah, no, um I think definitely models should support, or it is good to be good at this and important to be good at this. Yeah.
It's also a very niche case where we're better than AlphaFold 3, so we liked that one. To jump to the next point, which is we care about being able to template small molecules. This, as I said, is useful for a huge number of reasons. One of them is to enforce particular conformers if you only care about, say, ligand binding of a particular conformer. Other reasons are for virtual screening if you know what your structure of your small molecule looks like, or you know the structure of your protein looks like, and you only want to interrogate the docking. That's another use case for this. That's what we're showing on the plot on the right-hand side. So we're specifying here a conformer. when we provide no templates, we get a protein ligand interface LDT of 0.82, so that's unconditional. If we template the small molecule, that's what's shown on the left, where you see the small molecule conformer templated, and then the protein docks around it. We get that increases to 0.88, which is about the same as if you template the protein as well. And so we're not in none of this are we leaking the dock, but we are prescribing what either the protein geometry is, backbone only, or what the small molecule geometry is, and that actually improves our docking performance, which makes sense. And the picture on the bottom is just a particularly nasty molecule where we're able to correctly predict its conformation after templating. So this is sort of an obvious thing. Like, of course, if we tell the model what the conformation is, it will attend to it. But it ends up being quite useful in practice. And so we've seen uh, over and over again this be applicable because we have people say, hey, AlphaFold 3 does not predict my small molecule correctly. I know what it is. I want to predict it. This is a very clean path to do that without influencing the distribution, the prior distribution either with inference time tricks.
Can I also do atom level templating for the protein?
It can do atom level templating for the protein as well. Using it can do within a residue atom level templating. It can't do between residue atom level templating because the way we do atom level templating is with the user specified conformer.
So with the reference conformer.
Correct.
Oh,
Alternatively, I get because the templating occurs in two ways. It occurs through the template track at the token level, and it occurs through the atom level embedding track through the reference conformer. In theory, if you wanted to do atom level templating of a protein between residues, you could then atomize those residues and provide the template at the token level, which would also work because we train on that explicitly. A bit of a trick. Then jumping forward again, this is an example from our group of a phosphorylated peptide binder where we actually went and got a crystal structure for it. On this particular example, RF3 exhibited lower RMSD compared to the crystal structure than all other models available. And so that was exciting for us. This is the great work of Magnus and others that our model not only looks good in silico, but when we go to the lab and we go and get a crystal structure, which is not yet in the PDB, so we can be sure it's not leaked in any way, RF3 performs the best of all models.
can you
The
say, so can you say here how the phosphorylation binder was designed?
I think this was designed with a fine-tuned version of RFD2.
Cool.
The next thing I want to talk about is how we use these as filters for designs, which I think is probably one of the in our group, one of the most common use cases of structure prediction tools. And the most frequent way this is done is you have a sequence and you believe this sequence folds into a particular structure. You then fold that sequence to see if it does indeed fold into that structure. For enzymes, the way we often do this is we fold that sequence into a structure and we see whether or not key catalytic distances are preserved. And so I'm showing on the left here the active site of a serine hydrolase enzyme, along with certain distances labeled that are important for the activity. What you can do is you can take a bunch of putative sequences, you can fold them, and you can see which of those sequences best preserve the distances that you care about. So if you do this on this data set of serine hydrolases, which are a few thousand data points of sequences and their measured activities in the lab, we find that there is some degree of correlation between how well the structure adheres to those specified distances and the activity that we measure in the lab. Here, the probability of active is just a very simple decision tree based off of how many of these distances are being preserved in the recapitulated structure. And then the y-axis is the normalized experimental rate, which you can think of as enzymatic activity. So this result was exciting for us because it suggests there is some signal for being able to discriminate between active and inactive designs using structure prediction as a filter. The word of caution, which I wanted to throw as well, is that we actually have seen that in general for small molecules, these structure prediction tools are just terrible as filters. And for intuition for why this is, I wanted to talk a little bit about this case of steroids. So steroid receptors in the body are hyperspecific. If you have a steroid receptor that binds estrogen, it is certainly not going to bind progesterone, and the other way around. But these ligands are very similar. They differ by only a couple functional groups. And so, a brief way of exploring this would be to take a native uh, receptor, in this case, the progesterone receptor, and its native ligand, in this case, progesterone, and then also try to predict that same receptor with similar but non-native ligands that we know do not bind, in this case, estrogen and testosterone. And what you find when you do this is that these models predict them on top of each other, which is on the left hand, and they also predict them quite confidently, which are the IPTM scores shown on the right-hand side. This is a bit of word of caution, and it's that these protein ligand IPTMs appear to hold very little discriminative value for whether or not things actually bind and do not bind. And this is replicated with AlphaFold 3 as well in other models, is that these IPTM, these interface scores for protein small molecules, are wholly a artifact of whether or not we've seen a similar looking small molecule and similar looking protein pocket during training, and not indicative of chemistry. Because if this were indicative of chemistry, you would expect the model would predict binding with its native ligand, progesterone, much more tightly than it does with its non-native ligands, estrogen and testosterone.
So do you have an evaluation with some more targets than just this one case?
We haven't done a thorough evaluation yet. This is something which we, I think, have been aware of for a long time, but this is a clear manifestation of it. What we do have is we have around 5,000 designs that we ordered that had perfect AlphaFold protein small molecule metrics, and none of those 5,000 designs bound in the lab. And so this, for us, was a bit of alarm bells because if you order 5,000 designs that AlphaFold is two thumbs up, you would expect that at least some of them would work. And the fact that none of them worked means clearly something is wrong with the way that we are filtering designs. And these
Mm.
metrics are quite difficult. And more broadly speaking, the outputs of these models and the outputs from the confidence module look identical. So I do not have any conviction that any sort of bind, no bind model or affinity head trained with frozen structure prediction weights would ever be able to discriminate between these two because the model has no incentive during training to do anything other than predict these on top of each other. And I think that's an artifact of the way we train, and it's something that I think we are certainly thinking about how do we address this moving forwards.
What's cutoff do you use to say it's not successful for your designs that you ordered and you say none of them but actually bound? Like what do you mean with binding versus not binding?
I mean, this is in the wet lab. I don't know the details of the experiments, but it's
Okay.
in vitro. The assays we used did not perform. They looked like the negative control, let's say.
So you don't know if there's like a certain KD number that they use as the threshold for saying it's not binding.
No, I'm not sure of the details there. I think this was very interesting, and I think finding, but it's also not a surprising finding if we think about hallucinations in other space, like the language model literature. And so this is looking at an article that OpenAI published recently, which is called "Why Language Models Hallucinate," and I thought it was fairly uh, it makes it quite obvious why we would have this artifact that these models cannot discriminate between similar small molecules. And the reason is what they show on the left, which is the way we grade these models, is essentially through these binary tasks where you don't get partial credit and you don't get credit for saying, I don't know. Similarly, the way we evaluate structure prediction models, if I gave the model a small molecule and a protein and it wanted to minimize its loss, it should always dock that small molecule in the protein because we do not see any negative examples during training. The model has no way of saying, I don't know. The only way it can say I don't know is through the confidence module, but there's a stop gradient between the confidence module and the trunk. And so it essentially needs to learn how to be untricked by the core of the model, which is quite a difficult task. And then on the right-hand side, I think is a fairly instructive point, which is they show that O4 Mini, which is one of the prior OpenAI models, performs worse on accuracy than GPT-5 thinking, but it performs much better on these more holistic measurements, such as abstention rate and error rate. And the reason why they're emphasizing this is that if you narrowly focus on accuracy, which is the equivalent of narrowly focusing on structure prediction performance, like LDDT, you end up with a model that looks better on paper, but performs worse in reality. And so these thinking models are actually worse from a pure test taking perspective than the earlier models, but they're much better in terms of usefulness because they make fewer errors and they know when to say I don't know, which I believe like has some implications and ramifications for how we think about structure prediction. Because I think as a field, it would be helpful to think about how we can encourage models that say I don't know. And there's many ways of doing that, but it's not entirely clear yet what the best one is.
Yeah, I think this hallucination point is great, and but a bit separate from that. But going back to your small molecule binder design, what do you think about using Boltz-2 for Boltz-2's affinity prediction for distinguishing your binders and non-binders instead of using the IPTM?
So these are on in-house data sets. We did benchmark them, and we found it had less predictive power than AlphaFold 3 for us. I don't I would be curious to see a broader comparison on our own in-house data sets. The Boltz 2 affinity had less predictive power.
Then are you talking about distinguishing small molecules for the same target or about distinguishing different proteins for the same small molecule?
This was different proteins for the same small molecule.
Yeah, I guess the model was not trained for that. So um, it's like, right, it's trained with these or it's sort of contrasting losses for distinguishing two small molecules for the same target. And now, if we want to do better small molecule binder design, like where we design proteins that bind small
Yeah.
molecules, it would be interesting to like see what data and training procedures can we have to train an
Yeah.
affinity prediction model that can also distinguish between proteins.
I mean, I'd be very curious to see how the Bolt affinity head would do on this task, which is different small molecules for the same protein. I would be quite impressed if it could discriminate between these. It's possible. And again,
You I mean, it should be okay. Do you know the PDB, the CCD of estrogen and pro whatever of the
yeah,
chemical?
I can shoot them over afterwards, and I would be very
Okay.
interested in seeing the performance. Because what that would mean, which I think is significant, is these models are actually learning chemistry rather than memorizing patterns. Because in order to differentiate those apart, I think you truly need to learn chemistry.
Okay. Yeah, well, maybe can you go back to your slide? Um, uh, okay, you also don't have the protein sequence here.
Yeah, I'll shoot the sequence and I mean I have JSON inputs that we used. I can just send
Yeah.
them over to you.
Otherwise, it would have been fun for me to quickly run it, and then at the end of the meeting, I could say the
Okay. I mean, I can try to send them now if you want, but I think it might take me to
let's
find it.
Let's continue instead of holding things up. If anyone in the chat knows the sequence, then but we don't want to stop Nate from talking. Okay.
Okay, so then just thinking forward, what's next for structure prediction? A few things. I think as always, there's different trends in the ML literature that I think are exciting. Reasoning, I alluded to by talking about GPT-5 thinking versus O4 mini. Mixture of experts is another area we're looking into. We know this has been hugely successful within other domains. There's no reason it shouldn't also be successful within the biomolecular design domain. We're still interested in how to reintegrate physics-inspired features, especially as we see these models memorizing and not generalizing well. They don't adhere to chemistry. The question arises, how can we encourage them to better adhere to chemistry? And physics is a natural way of doing that. And the third point is I think we really need to change the way we evaluate in order to. I think the Bolts 2 was completely a step in the right direction and helped pave the way for this, which is we really care about structure prediction models that can discriminate between binders rather than structure prediction models that naively optimize towards arbitrary metrics such as LDDT, which is just not representative of the reality of the way these models are used in practice. All right. Any questions about structure prediction world before we?
Yes. So why do you think a mixture of experts are interesting here?
Uh, there's this finding that we have and that others have as well, which is that there's very little transfer learning between modalities. So getting better at protein DNA doesn't really make you that much better at protein RNA. And getting better at small molecules doesn't necessarily make you better at nucleic acids. And this suggests to us that the model is using different parameters, different portions of its parameter space for each of these tasks. When you have such a situation, I think a fairly natural approach would be to use a mixture of experts where you could hypothesize that one would become really good at understanding DNA and the way it interacts with proteins. One would become really good at small molecules. And it seems like a reasonable way of increasing your number of parameters and without uh, increasing the likelihood of overfitting your data set. And so we haven't fully explored this yet, but we're hopeful that it will ultimately lead to some greater performance, which it clearly has in other fields. So like I think the set of orb models from Facebook similarly uses mixture of experts in a analogous task, but on a much smaller scale. And then across all of the language model literature, it's taken the field by storm.
Yeah, so my understanding of mixture of experts for language models is that we want to be able to memorize more facts faster. And yeah, there we have essentially infinite training data, right? We never we don't complete a single epoch. And yeah, then maybe it can make sense that you want to be able to pick up faster on facts and memorize. But now the thing that you said without overfitting, I don't quite see how a mixture of experts
Mhm.
help
Mhm.
with not overfitting.
No, no,
Now,
yeah, that's a fair point. I guess to your earlier point about trading data, so we already do not see all of our trading data. We have enough distillation now that we can train for 20 million examples and not see all of our training data. So it hasn't always been the case, but I think we are now that we have sufficient scale such that it may make sense to use a mixture of experts type approach. To your second point, I think you're absolutely right. Like one thing we have to be very aware of is if you end up with an RNA only expert, that RNA only expert would immediately memorize the PDB. And so we have to be extremely cognizant that the way we sample, we reroute between the experts doesn't enable it to overfit in this manner, and that we make ample use of our distillation data to encourage broader generalization.
yeah, okay, and then for reasoning, right, we can or sort of doing some inference time search for your better predicted, maybe worse predicted structure, it is then again that we need a good confidence model, or if we're talking about design, a good binder from non-binder distinguishing model. Do you see any other ways how inference time reasoning can help us?
I think it only really works with some form of confidence metric that you trust. But if you think about the antibody case, it could be a good candidate where the model could run the trunk and then it says, do I think this is good or not? Do I want to run again? And you continue iterating, which is the equivalent of thinking in this case. So I'm using recycling as a term for thinking. And you continue iterating until you arrive at something you are happy with, and then you denoise into a structure. And so I think that is a natural way of thinking about thinking within the context of protein design. I think it would certainly require some degree of train time as well. It cannot be done exclusively at inference, but in most of the language model literature, it's done in post-training. So it wouldn't be as computationally prohibitive as some of the other tasks and topics we've talked about, because it could be performed off of a pre-trained model that already has a good understanding and foundation of protein structure. And then you encourage it to cycle more recycles when it wants to further refine a prediction. I think the other interesting point that could be made is you enable the model to not only choose whether or not it wants to continue recycling, but also to see all of its previous recycle outputs, such that it would encourage it to explore more diverse a conformational space. And what I mean by this is what you see when you perform many recycles, so that each recycle oftentimes is the same thing or very slight modifications of the same thing, where you actually want it to be trying out different docks and then deciding whether or not that dock is good and exploring over a broader range of space. And I think how to do that, we have most certainly not figured out, but I think it's an interesting idea to pursue.
Cool.
Great. So keep rolling forward here, I'll talk briefly now about design. So I talked about AtomWorks, which is this underlying framework that we developed. We've used AtomWorks to train several models. One of them was Rosetta Fold 3, which we showed. And another model is RF Diffusion 3, which shares a huge amount of code with Rosetta Fold 3, but we essentially adopted the framework and the architecture to be used for design as opposed to protein structure prediction. I don't want to talk too much about this now because I think this will be a future topic in and of itself, but want to give a brief preview because I'm excited by the work that so many great people in our group has done. So high level RF diffusion 3 is an atom level, it's an atom 14 model that enables a diverse range of biomolecular tasks. And so we're able to do enzyme design, we're able to do DNA, RNA binder design, we're able to do PPI design, this broad range of different biomolecular tasks. And we're able to perform them at far greater speeds than before and with higher levels of success. So we're showing on the left-hand side, this is co-diffusing DNA with a DNA binding protein. And we actually went to the lab and experimentally validated that these proteins do indeed bind DNA, which has been a grand challenge for many within the de novo design space for some time. So we're very excited by this. Similarly, on enzyme design, the fact that RF diffusion 3 is natively all atom, as opposed to operating in the space of frames and translations, has enabled us to significantly surpass the previous benchmark set on the enzyme scaffolding task. And so this is if you give a given set of tip atoms, which you believe are catalytically relevant for your atom or for your enzyme, can you scaffold a backbone that accommodates those atoms? And which after refolding is retained in the outputted structure from AlphaFold, from Chi, from RF3, what have you. We find that this atom level conditioning enables us to significantly exceed the prior benchmarks and then make also the point that it's way faster now because the architecture is far simpler. We don't have any equivariance. We don't have any SA3 transformers. We have a very small trunk, only a couple pairformer layers. So it's comparatively, compared to RF3, much, much, much faster. So we perform better, not only on absolute pass rate, but if you time or GPU hour normalize the pass rate, the numbers look quite a bit better. We've already seen that we're able to go after enzymes now that previous methods were entirely unable to design against. And so we've had active enzymes designed in our group that were tried repeatedly with RF diffusion 2 and were never successful. And we've found now that with RF diffusion 3, we've been able to design active enzymes for these difficult targets, which is super exciting. Great. So that's all I had. Happy to answer any questions, and then I'll send you the sequences afterwards, Hannes.
Very cool. Yeah, then should we maybe try to have you quickly send the sequences while people can go into the depth of their questioning and try to see if they have some questions, some thoughts, and if not, then I start running.
It would be cool to do this live. Let me see if I can find the sequences.
Yeah, so friends of the sun, if you have any thoughts, questions, let us know. Feel free to unmute. Uh, yeah.
I just had a little detailed question on the all atom uh,
Yep.
or the all atom templates. So you basically have a flag that you give in the reference conformer that says these are like ground truth coordinates, so really pay attention to this, and then you just sample that sometimes during training. Is that
Yeah,
what it
so
is?
we provide templates in two ways. One of which is we call the ground truth conformer, which is exactly what you described. Um, we actually just provide it via a different track. So when we provide the ground truth conformer, we also provide a normal conformer, but we have this separate track which we exclusively use when we're providing the actual ground truth, so that the model can devote a set of weights to exclusively copying over things that appear in this other track, which is usually zeros, except for when we provide the ground truth conformer, in which case it hopefully just copies it over. So that is the first way we template. The second way we template is at the token level, which is more typical of how AlphaFold 3 uses templates. But instead of providing templates from homology, we provide slightly noised versions of the ground truth. So we typically add a small amount of noise to the token coordinates, the representative coordinate for each token, and then embed those as a histogram, which we provide to the model. We use this noise such that it has some degree of flexibility. We haven't exposed this yet, but in theory, you could specify the amount of noise that you want to add to your template, which would enable the model to understand how closely to adhere to a given template. And we're excited because we originally added this templating feature in fine tuning, and the model adhered quite well. But in the run 10 that I showed earlier, which is now quite a bit better than RF3, and we'll probably release it this week or next week, the templates are adhered to much more strongly because we trained from scratch with it.
That's great. And the token level thing can be across chains and can be any.
Yes, the token level thing can be across chains. The token level thing can also, it can be, as you say, interchain as well. So you could specify not only what a chain looks like, but the relationship between two chains, because we do, with some very small probability, provide that to the model. Whenever we do provide that knowledge to the model, the gradient updates with respect to those parameters are huge because it provides a lot of information. If it knows how two chains dock, so we have to be quite careful about when we provide that feature.
Cool. Thanks.
Honest, I think the sequences are buried. I'll have to send them to you afterward.
Sounds good. Sounds good. Yeah, someone else unmuted, but that might be resolved. Then I would say thank you, Nathan or Nate, and yeah, we look forward to the release, I guess, that you alluded to, like the new folding model. That will be super cool. And with that release, do you think you'll also have some more concrete comparisons with AlphaFold 3?
Yeah, I suppose we'll probably have to do that.
Yeah. So do you think it'll close the gap completely to AlphaFold 3?
I do not. I think we need the remaining 20 million distillation examples and maybe some data set sampling, but I'm hopeful that others in the community will close that gap as well. I'm hopeful of that.
Okay. Well, cool. Then thanks a lot for the chat. Like, thanks a lot for the explanations. And if you have any last words, now's the time.
No, please shoot me a note. I'll put my email in the chat if anyone has any further questions.
Okay, good. Then thank you, Nate. Thanks everyone for joining, and see you around, Friends of the Sun. Bye.
Thanks, Hannes. Bye-bye.